Good evening, ma'am. Good evening, ma'am. Hello, ma'am. Good evening, ma'am. Good evening, my dear students. You are all warm. Welcome to the another biological session. So last class, what we have discussed already, you know about, okay, genetic engineering, the part 2, isn't it? So last class, what are the applications in medicines? What are the medicinal useful recombinant product is there? So all those things already we discussed. Okay, then we saw that engineering of insulins. Insulin also we saw it in the slightly. So today we are going to discuss about the remaining part of the genetic engineering insulin, how they will produce inside in the laboratory aspect. That things also we have to discuss. So you know about that insulin. Insulin is a very important hormone secreted by the Langerhans, liver, pancreas, Langerhans of alpha and beta and gamma. There are 3 cells out there. Here, beta cells produce insulin. Okay. First of all, you should know what is meant by insulin. What is it is also called as, what is it? Hypoglycemic hormone. What is meant by hypoglycemic hormone? Glucose level, it will be reduced when the blood glucose level is increases. Okay. So, automatically the liver, pancreas, beta cells secrete the insulin hormones. The insulin hormones act upon it. As a result, what happened, the blood glucose molecules converted into glycogen. Okay. And also stored in the liver. It is called as animal starch. It is called as animal salts by the help of insulin. See that one. Insulin used for diabetes. Suppose the production of insulin secretion is stopped by the beta cell. What happened? The glucose molecules excreted through the urine. So that condition is called as diabetes. Depends upon the concentration of the insulin molecules. It may be secrete, mild, moderate, severe cases. There are 3 types have been reported. Okay, due to the insulin deficiency. So that cases are the conditions we have to introduce intravenously insulin produced by the laboratory aspect, otherwise called as commercial aspect prepared one. We will use it for the remedy. Okay, see that one. The insulin used for the diabetes was early from pancreas of the slaughter cattle. Slaughterhouse already we discussed. Okay, so slaughterhouse, okay, cattle and pigs, and caused some patients to develop allergy. Suppose we are taking insulin of some animals. Okay, the animals insulins also act as a human insulin. So we collect the insulin from the slaughterhouse. Okay, and also we are supplementally we are giving. That time what will happen, so some of the person develop allergy. It will not, okay, it will not be a good result. So that time what they will do, they use commercially prepared one, laboratory aspects. Here, the insulin consists of the 2 short polypeptide chain, already we discussed. There are 3 chain is there, but 2 chain is very, very important one. That's why they have told alpha chain and beta chain, and simply called as, A chain and B chain and C chain. Okay, C chain is also there, C peptide also there. First of all, you should note it down. What is the structure of insulin? The insulin consists of 2 short polypeptide chains. The chains A, the chain B, and chain C. Okay, that are linked together by disulfide bridges. Already we discussed what is by disulfide bridges. That things also already I showed. See that one. So this is the entire structure of the insulin molecule. Here it composed of a nucleotide. So this is considered as each and every bit is considered as a protein molecules, otherwise called as amino acids. It composed of how many amino acids already we discussed. Okay, so 50 Okay. See here, how many chains are there now? See that the green color given as A chain, the dark black color is here C chain, the orange color is here B chain. You have to tell us the A and B and C chain. Here, the C and A, it will not connect. So, with the help of the disulfide bond. Here, A and B only connected with the disulfide bridge. This is considered as a disulfide because sulfur connect these two component. In between, there is a bond made up of sulfur, sulfide, simply called as disulfide because two molecules involved in this bridge. So it is called as what is a disulfide bridge. That things has given here. See that disulfide bridge. In mammals, including human, insulin is synthesized as a prohormone. Already we discussed about that prohormone. See that one? This is the A chain, this is the B chain. Both are connected with the disulfide bridge. Addition with this, there is a C peptide is enhanced, or what is the extension is also that. Suppose if it is extension present in it, that hormone is considered as a proenzyme, proinsulin. Okay. During the maturation of insulin, what happened? The extension is completely nil or removed or absent. Okay. See, don't forget, my dear student. This is very, very important. This is the entire structure of the which one insulin hormone. It contains 3 chains: A chain, B chain, and C chains. The A chain and B chain connected with the disulfide bridge. Suppose the extension is there, okay, it is considered as a proenzyme. The mature enzyme, what happened? That extension, it will be completely absent. That is called as C peptide. See that one? In mammals, including human, insulin is synthesized as a prohormone that contain extra stretches. Extra stretches, that is very, very important. There is a extension is there. That is called a C peptide. The C peptide is not present in the mature insulin and removed during maturation into insulin. So the insulin before that insulin is converted into insulin. Okay, so that is very, very important. How it will be? So due to the absence of C peptide. That is very, very important, my dear student. Don't forget. Suppose the presence of what is that? Peptide. It is considered as a C peptide. Don't forget it. C peptide. It is considered as a proinsulin. Absence of C peptide. It is considered as a what is that? Insulin. Otherwise called as mature insulin. Okay. Okay. Absence of insulin. Absence of C peptide. Presence of C peptide. You know the difference between these two. See the entire structures. Already we discussed yesterday. Here also, once again, I repeat for you. The A chain consists of 21 amino acids and B chain consists of 30 amino acids. And C chain also has 30 amino acids. Total 81 amino acids there. Sometimes they may ask the question in NEET point of view. The insulin component contain, insulin hormone contain, how many chain is there? A chain containing how many amino acids, B chain containing how many amino acids, C chain containing how many amino acids, and B and C are both are same. A chain, it has 21 amino acids. Okay, so not only that, and also the insulin gene has 153 nucleotides. See here, you just calculate from here to this end of this one. This is a carboxylic end. This one is called amino end. You calculate it, how many nucleotide present in 153. Don't forget, this is very, very important. The 153 nucleotide is also present in it, and also the chromosome number 11. Okay, we are having 46 chromosome and 23 pair of chromosomes. From this 11th chromosome responsible for the production of insulin. So the 11th chromosome is responsible for the diabetes. Okay, the insulin is a dimer of an A chain and B chain. Dimer is the two compounds linked by the disulfide bond. It is composed of 51 amino acids because these two is added, you will get 51 amino acids. with the molecular weight of 5,808. 5,808. This is very, very important, my dear student. Don't forget. See it here. So this is the entire structure of the insulin molecule. It consists of A chain, B chain, C chain. Right. A chain containing 21 amino acids. Count it here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21. So here he just calculated 30. Here also 30. And A chain and B chain connected with the disulfide bridge. Okay. Total how many amino acids are you calculate it? Okay. So 81 amino acid sequences there. 81 nucleotides. Particularly A and B 51. Here 30, here 21. Okay. Disulfide bridge. Already know about. C peptide. Addition of this one, C peptide. Absence is called as mature insulin. Okay, that things is very, very important, my dear students. So many times they were asked the question. See that this is called as pro-insulin. See here A peptide, B peptide. Okay, after that what happened? No, free C peptide. This is considered as a free C peptide. So as a result, you will get the mature insulin. Okay, so this is the structure of the insulin molecules. When there is these molecules are introduced, where there is a production is takes place in commercial aspect, that things we are going to discuss. 1983, Eli Lilly, an American company, prepared 2 DNA sequences corresponding to A and B chain of the human insulin. Who is that? 1983. Okay, Eli Lilly and the American company produced the A and B chain of the human insulin and introduce them into the plasmids. PUC, very, very important. It is another form of plasmids present in the Escherichia coli, otherwise called as E. coli, to produce the insulin chain by biotechnology methods. Okay, don't forget, my dear student. Here the 3 question is that who first introduced or prepared? 1983. Eli Lilly, American company. 2 things. How many chains is there? A and B chains of human insulin. A and B chains of human insulin. Which plasmid is responsible for the PUC? PUC. Escherichia coli to produce the insulin chain. This chain consists of A and B were produced separately, extracted and combined by creating disulfide bond to form a human insulin. What is that meaning? You know, these 2 chains are produced in different companies, different areas. After that, it will be united with the help of the disulfide bond. Disulfide bond. Don't forget. Okay. Disulfide bond. SH bond. Okay, so disulfide means you know about the 2 molecules of SH bond. Okay, so as a result, what happened? Human insulin formation takes place. These are all done in the laboratory aspect. See this one. So here there is a insulin A, insulin B. These are the different types of molecules also separated. Insulin A, insulin B, insulin C, like that. It is depends upon the component. Here there is a plasmid already in our blood. The promoter is the LAC-Z gene. Here the LAC-Z gene. So the LAC-Z gene is essential for the human insulin production. Already we discussed. The insulin gene A subunit. Here insulin gene B subunit. That is means this insulin production foreign DNA molecule isolated from the E. coli, but at the same time the sources will be differ and also introduced into the E. coli. This 2 is a coast. After the cutting and also ligation process also done, after this we will introduce into the coast. Inside the coast, what happened, you know, already in our body, it also undergoes the multiplication process. It will produce particularly the insulin A subunit containing proteins. Here B subunit proteins. Okay, so these are all very, very important concept. What is the next one? Development of genetic engineering vaccines. So how the genetic engineer vaccine preparation takes place? What are the diseases already? There are so many diseases have been reported. Nowadays, we are facing so many problem also. See that scientists are now applying the technique for genetic engineering to the development of vaccines by inserting one or more of the pathogens gene into a non-pathogenic organisms. What is my pathogen? Pathogen is nothing but the disease causing organism. Only they produce the disease only. Such genes are introduced into the non-pathogenic, those who are not affected by the disease, that organism as a host. We are introducing the pathogen gene into the non-pathogenic genes. As a result, what happened? It produced some diseases. Take an example. Any one of the foreign particles. I will show you how it will be. Take an example. Okay. This is a human body structure. Imagine any one of the cell. Take an example. Okay. So this is considered a non-pathogen. This is a considered a human body. Okay. Human body. Here the foreign particle, take an example, any one of the virus, any one of the virus. This is called as alien species or foreign, alien. Okay. Alien, okay. Alien gene, DNA molecule. When it enter into the body, when it enters inside the body, what happened? On the surface of the human body, what happened automatically? Sometimes it may be enter inside. Okay, enter inside. So after the entry, what happened? No, automatically it also react with the host DNA molecule, host DNA, isn't it? Host DNA molecule is that host DNA, isn't it? It is also incorporated with the host DNA. This is the viral DNA, viral. So DNA, and this one is considered as a host DNA. Okay, host DNA. After the incorporation of the viral DNA as well as the host DNA, what happened? All the cell activity controlled by viral DNA only. Take all the activities. Okay, vital role. Viral DNA only. As a result, the mRNA, tRNA, RNA, all are synthesized the protein molecule for what is that? Viral DNA proteins. So inside what happened? That protein is called as what is that? Antigen. Antigen. That protein molecule is called as what is that? Antigen. Okay. Antigen. So automatically when the antigen molecule present in your body, automatically it express their symptom, whatever it may be. Okay. Symptom. Okay. Symptom. So as a result, the host suffer by an improper function. That is very, very important. So we went to doctors. So doctors provide, what is that? Doctors provide the supplementation, isn't it? Supplementation. So the antigen already in our body is simply called as antigen. It is a viral protein. Okay. And then that time what happened? They will give the antibiotics. Antibiotics. So as a result, the antigen antibody complex formation takes place. So the disease will be completely removed or prevented. That is the reason. So the antigen is completely, what is that? Denatured. That is very, very important, my dear students. Okay. So we will pass on to the, okay. See here. So this is the important point. Okay, so that things we are discussing here. See that the scientists are now playing a technique genetic engineering to the development of vaccine by inserting one or more pathogen gene into the non-pathogenic organ. Scientists have produced a successful preparation of hepatitis B vaccine. So here what they did, the hepatitis B virus is injected into the what is that? Non-pathogenic organisms. As a result, the pathogen inside, what they will do, already we discussed in the previous slide. They produce the antigen. Okay, from that one we can produce the hepatitis B vaccine. That is very, very important, my dear students. Okay, see, this is the hepatitis virus, hepatitis antigen producing genes. It is also isolated from the particular virus. It is also incorporated with the plasmids molecule isolated from the bacterium, extra chromosome body plasmids. So both are combined together. What happened? A recombinant process also done. After the recombinant process, what happened? We have to introduce into the host cell. Automatically, what happened? No, this gene produced within the E. coli host. So you will get the which one HB antigens. Okay, the antigen producing component. It can be commercially prepared and also what is the marketed. So this is the entire technique in simple way I have told, isn't it, my dear? So this is very, very important. Okay, next so concept booster. First you should note it down. How many vaccines is there? What is by DNA vaccine? What is by DNA vaccine? What is by RNA vaccine? Some of them are protein actor also act as a vaccine. See, the DNA vaccine, the most recent vaccine technology involves the development of DNA vaccines. Okay, DNA as a nucleic acid, it is also used for the production of vaccination process. That's why it is called as DNA vaccines. The DNA vaccines works by injecting the DNA coding for a particular segment of the disease causing organism into the muscles of the person being vaccinated. Okay, the DNA becoming incorporated into the muscle cell of the recipient and cause those cells to produce the protein for which the DNA codes. This is one of the method. Okay, what is the meaning? DNA, which one is responsible for the production of vaccine, that is identified and isolate and also used as a hybridization technology. After that, you will get the DNA containing vaccine because it is also isolated from the DNA. That's why DNA producing vaccines. See, DNA vaccine have some, what is the advantages over the types of the vaccine preparation. This is also very, very important one, my dear. So take an examples. We also did, okay, Covaxin, Covishield. That things, what is that meaning? The particular pathogenic part, whether it may be DNA or RNA or a body part of the virus. Which one, COVID virus? That particles is isolated from the original pathogen and also inactivated by some specific technique they were used. Physiological changes or chemical changes which occurs. After that, it is also tested by many numbers. After that, it will be introduced into the market. Covishield, Covaxin, or what are these things? All these are all are containing COVID virus particles, whether it may be a nucleic acid or protein molecules, the outer hooks like structure, spike. See that one. DNA vaccine have many advantages over the other type of vaccine preparation. First of all, no risk of infection by the pathogen. First one. Second one, no risk of illness from contaminations of the vaccine. Second one. A long-lasting immune system response. Third one. The ability to administer a vaccine for a variety of disease in a single shot. Okay, these are the advantages prepared by the DNA used as a vaccine. Okay, this is very, very important, my dear students. Don't forget. Sometimes they were asked the questions. Okay, okay. So go to the next one is transgenic animals. You should know, so many animals have been reported. So many animals also under the research. Okay. Here you must note in which animal first introduced. What are the types? What is the habits? Okay, all those things you should note it down. This is very, very important, my dear student. This sector or this part, many questions are arrived. See that transgenic animal. Transgenic animals means both different. Transgenic. Trans means different. Genes also isolated and united. Okay, the technique already you know about biotechnology or whatever tissue culture or whatever it may be. Okay, that methodology we are using which one you know? We are producing a new variety of animals, new gene of animals. Okay, see that one. Transgenic animals serves as an important model for clinical research. Clinical means disease. Okay, clinical research. Don't forget, my dear. As well as a bioreactor in pharmaceutical manufacturing sector. This is very, very important. Suppose if you are introducing a new vaccine, it will not directly introduce into the human being. It will not introduce in the human being because suppose if it is lead to death, you know, okay, how many humans it will die. That's why before the introduction of the human beings, what they will do, you know, before checkup 100 and 1000 times they check with the animals, laboratory animals. Take an example: guinea pigs, mice, pigs, rabbit, sheep, cow, fish, and so on. So in there, monkey. Okay, so those who are all are mammals. After that only it will introduce in your body. Okay, see here. So pharmaceutical manufacture sectors, the term transgenic was first coined by Gordon and Rodil in the year 1981. This is very, very important, my dear student. Transgenic, the term was introduced by the name of the scientist Gordon and Rodil in 1981. Okay. The transgenic rats, rabbits, pigs, sheep, and cow and fishes have been produced, although over 9% of all existing transgenic animal are mice. That is very, very important. So many animals have been identified. Some of them are, what is that? Positive result, we got it. And some of them are negative result, we got it. Negatives are destroyed. Positives are analyzed. Okay. See that one. What are the example has given? You must here note it down. 3 point is very, very important. Transgenic animal mostly involved in the clinical research and pharmaceutical manufacturing. First transgenic, the word first introduced by the term was coined by name of the scientist Gordon and Riddle in the year 1981. What are the animals? How many percentage? 9%. Okay, it's also existing transgenic animal on mice. See the next one. The first transgenic animal. First notable biological mode, so superior mouse, otherwise called a super mouse, were produced in 1982. Sometimes they may ask the question, which one is the first transgenic? What is that? Animal, like that they may ask the question. Goat, sheep, mouse and mice, like that they may ask them. Don't forget mouse. Okay, in the year 1981. There were mice introduced with the growth hormone genes. The mice induced with the growth hormone genes. Transgenic animal that produce useful biological product can be created by the introduction of the portion of the DNA that code for the particular product, such as human protein. Okay, that is very, very important. Transgenic animals here, what they did, you know, they isolated the human protein molecule DNA that is also introduced into the mouse. Okay, so the research is also data. How? See that in 1997, the first transgenic cow, Rosie. Don't forget, my dears. This is also very, very important. Okay, produce human protein enriched milk 2.4 gram per liter. The milk containing, very, very important, my dear student, the milk containing alpha-lactoalbumin and was nutritionally a more balanced product for human babies than the natural cow milks. So this is the very, very important points, this one. Okay. Sometimes they ask the questions. Which one, you know, the cow Rosie, the match the following is also they ask the questions. The Rosie cows, it's contain which human alpha-lactoalbumin, okay, gene responsible for the production of alpha-lactoalbumin. It is similar to that of the natural cow milks. Okay, that is also very, very important, my dear. Next, we will pass on to the next one. This part is very, very important, my dear. Don't forget, once again, repeat for you. In 1997, the first transgenic cow, Rosie, produced human protein enriched milk. Human protein, don't forget. Which protein? Human protein. Okay, how much is that? 2.4 gram per liter. That is called as alpha-lactoalbumin. This is the nutritional substance. That protein molecule is called as, what is that, you know? Alpha-lactoalbumin. Don't forget. Okay. So go to the next one. Transgenic sheep. The female sheep having human anti-hemophilic factor 9th gene. The transgenic sheep is also, it contains a female sheep having the human anti-hemophilic factor. It is essential for the blood clotting mechanism. Already we studied how many factors responsible for the blood clotting factors. 14 factors responsible. Particularly, the 9th factor is responsible for the anti-hemophilic factor B, hemophilia B. Okay, this gene is their genome have been produced. They secrete human clotting factor 9 in their milk. So when we are consuming this type of milk, what happens? You know, we cannot suffer by anti-hemophilic factor, blood clotting factor. You will get it. That is very, very important, my dear students. Okay, so see the next one. So these are the sheep. See that donor. You have to take and incorporate it, and finally fuse the cell, and also that blastula stage. It's also once again entered into the, which one, you know, surrogate mother. After that, you will get the lamb. So it has these two characteristics. That is very, very important. These two combination of genes. These two combinations, we get it in a single, what is that? Sheep. That is very, very important, my student. So see that this is the sheep success. Okay, it has a fur for, what is that, woolen or something. We will make some commercial purpose, and also milk also, and also their meat. That things also we were mostly found in where, you know, hills rates, Himalayas, Himachal Pradesh, Kashmir, and that area it will be live. And the next one is to transgenic goats. It is also similar to that of the sheep. It's a variety of sheep, isn't it? It's a goat. The goat have a human gene that code for the blood clot dissolving protein. Blood clot dissolving protein. Don't forget, my dear student. That is called as what is that? Tissue plasminogen activator. Simply called as TPA. Where it present transgenic code it contain. Okay, which gene? Blood clotting dissolving protein. Okay, so tissue plasminogen, that protein is called as tissue plasminogen activator, have been formed. They secrete this protein in the milk. So it is very, very important when you are consuming this type of milk. What happened? No, we will not suffer. Which one, you know? Blood clotting process, hemophilia in nature. Okay, proteins. This is also very, very important. So you have to take a more amount of milk in this area where this type of goat milk, when we are consuming, what happened? You know, you will get the plasminogen activated protein molecules. As a result, the blood clotting is also prevented. That is very, very important. See this one. So this is the human gene. This is the egg donor. Both are come in contact close together with the process called as, what is that? Hybridization, biotechnology, and genetic engineering, and also insert to the female. You will get the milk. That milk it contain, what is that? That TPA protein molecules activated blood clotting mechanisms. Okay, it prevent the blood clotting things. See that one. This is also another one. Okay, these are the two types, different types of protein molecules also produce. Okay. So go to the next one. Transgenic cow. the cows containing genes coding for the antibacterial human iron binding protein, that is called as lactoferrin, have been produced. Transgenic cow produce lactoferrin. It is nothing but the antibacterial human iron binding protein. Ferrin, you know very but iron, isn't it? Lacto is nothing but the protein molecule. So the protein molecule contain what is that, iron containing protein. Take an example, hemoglobin is also there. Hemoglobin is also a iron containing protein, but it acts as a transport protein. But here, cow milk containing antibacterial human iron binding protein is called as lactoferrin. Okay, this is also a very, very important one. And second one is transgenic pig. Okay, pig labeled as a super pig. Why? Normally pig used for the, what is that? Meat. Okay, and some of them are used for the insulin preparation in commercial oriented. Okay, having human growth hormone gene, that is very, very important. What is the gene present in here? You must note it down. Transgenic pig or transgenic cow, transgenic sheep, transgenic mouse, or fish, whatever it may be. Here you must note it down. 3 point is very, very important one. Which gene is responsible? Having human growth hormone gene. Okay, what is the use of that one? Okay, example. These three must note it down. Here, okay, first of all, you should note it down. Transgenic pig also is called a super pig, having human growth hormone gene have been formed. Such pigs grow a little faster and have greatly reduced subcutaneous fat. This type of pig always is not, what is that? Fat content. It will not be a fat. It is the lean body mass like that. Subcutaneous fat completely reduced. So they can thus provide the leaner pork with lower fat content. That is very, very important, my dear. What is the two purpose? Two things is there. One is called as, what is that? Human growth hormone secreted by the super pig. And not only that, that lower content fat also present in the pork meat. These are the two things, pig meats. Okay, these are the purpose they were used for the commercial aspect. And another one is very, very important. Okay, another one is very, very important points. See here, xenotransplanters. Xenotransplanters. What is the meaning of xenotransplanters? Geno means, suppose take an example, any one of the animals organs we may transplant into the human being. Take an example, heart is failure. What happened? Sometimes they may took from monkeys. Okay, kidney failure. Sometimes they took from where? In the goat. Okay, liver, pancreas. Likewise, we will take the organ from any one of the animals and also transplanted into the human body is called as xenotransplanters. The xenotransplanters are the transgenic animal designed for the use of the transplantations of animal cell tissue and organ to treat or mitigate human disease. Mitigate. Okay, mitigate means cell related one. Okay, the most commonly considered animal for xenotransplant is a pig. So this is very, very important, my dear students. Maximum they took from any one of the organ from where, pig. Pig are the most physiological similar animal to human. That is very, very important. Xenotransplanters, you must note it down. Pig is a important one. See the structure that can easily identified. Xenotransplantations. This is a genetically modification donor of the animals. Whether heart, kidney, liver, pancreas. These 4 organs we can easily change. Whether affector or any other defect in human beings, we can change it. We can replace it. That is very, very important, my dear students. That condition is called as xenotransplantations. Mostly Which one is similar? Human organs are similar to the pig. Don't forget, my dear students. This is very, very important, and important questions also arise from this part. Next one. See that xenotransplantations. Exogenitalia. So here we have to take a cell, okay, and also grow into the pig, and the removal of that particular organ and transplanted it into human body. This is called a xenotransplantation. Otherwise, called as we have to take a organ donor. This is the donor to recipients. Okay, the particular organ, particularly kidney. In these cases, what happened? You have to isolate from the DNA molecule from the person and introduce in the culture medium. After that, it will be introduced into the pig in vitro. So inside what happened, it will grow as a kidney cell. Okay, we have to isolate from the cell from the human body of the kidney. Then it is introduced into the biotechnological changes also occur, physiological and chemical changes which occurs inside the lab. After that, it implantation into the pig. After sometimes what happened, the well-developed kidney also we can get it and also transfer to the. This is called as exogenesis. Xenotransplantation, directly we can change it. Exogenesis means, exo means outside. Take a cell from the human body and culture and introduce the pig. After well-developed organs, we can transfer into the human body. These are all are called as what is exogenesis. Okay, this is very, very important, my dear. And then next one is called as transgenic poultry. What is meant by poultry? Poultry mostly used for two purposes only. One is egg, another one is called as meat. Okay, transgenic poultry is also two things is that egg laying capacity, it will be increases, at the same time the muscles also, okay, the weight, and not only that, and also some of the fat is also removed. Okay, see that one. Transgenic poultry animals were developed to increase the healthliness and nutrient quotients of meat and other product. What is meant by other product? Other product nothing but egg. But in this category, when we use the transgenic poultry, what happened? In the egg is also, number is also increased, at the same time less saturated fat, lactose levels also less. Okay, they have no good quality meat and produce egg having less cholesterol and fat. Already you know both. Okay, taken examples. Okay, so any one of the egg, take an example, say any one of the egg. Okay, egg, isn't it? This is considered egg. Okay, egg. The center, already you know about center, there is a, what is that? Molecule is there. What is that? Egg yolk. This condition is called as egg yolk. And the both the side, what happened? There is a, what is a structure is like this. There is a extra structures. And that outermost layer is also there. Outermost layer. What is that? Outermost. The inner also, there is a layer is there. It is also covered. Okay. It is also a covered one. So this one is called as, what is it? Calcium carbonate, outer cell, simply called as HCl cell, egg cell, isn't it? It is made up of calcium, okay, calcium carbonate, okay, carbonate. You know the difference between calcium carbonate, okay, egg cell. Calcium phosphate, it is used for the teeth. Don't forget, my dear students. Okay, calcium phosphate here, C and A, calcium and A, phosphate in this category. It is essential for the formation of teeth and bone. Okay, but the outermost layer of the egg cell is made up of which calcium carbonate. Inside what happened, it is inside, there is a small molecule is there. Okay, inside there is a molecule. So this one, this part. Okay, so this part, what is that? The simple protein molecule. This simple protein molecule, colorless, that is called as albumin. Okay, albumin. Okay, otherwise called as simple protein, albumin. This is the center. There is another part is there, that is called as egg yolk. Egg, what is that? Yolk. O-U-O-L-K. So here, what happened, the lipid and phosphate concentration, lipid, otherwise called as lipid, as well as the fat concentration will be very high in nature. That is very, very important. In this part, lipid concentration is very, very high in nature. It is reduced by the method. Okay, transgenic poultry product. What happened, it will be reduced. Okay, see that. That is very, very important, my dear students. See this one. See this one. Transgenic poultry. Okay. Here they have good quality of meat and produce the egg having less cholesterol and fat. This is very, very important. Okay. Number of egg, it will be more. At the same time, that meat will be very less fat contained. Okay. It will be produced by the transgenic poultry method. See that one. The method of creating transgenic bed. Okay. So these are the so many techniques. See here. So these are the important things. Second one is also very, very important. So this one. Okay. The A and B and C. So these are the development method. Once the embryo stage is there, they introduced some injections. They used the injection. As a result, what happened? The embryo is completely male only. How many? So normal, this one already in our report, this one. Okay. So this is the one part, this is another part. When the male and female come in contact close together, as a result of formation of zygote, that time we'll get the progeny similar to that of the parent, isn't it? But in this category, we will get it only one type of parent only, male only we will get it. Okay. Sometimes it is female only for the egg laying process as well as the meat productions. Okay. These are all are the important only for the commercial aspects only, meat and egg. Okay. And then next one is called as first transgenic pre-made. Don't forget, my dear students, this is very, very important. So many times they were asked the question, which one of the following? Okay. The first transgenic pre-made. This was first introduced by the name of the scientist called as Dr. Gerald Saturn. Create the first genetically modified rhesus monkey called as Andy. Okay, don't forget, my dear student, this is one first prepared, which one are transgenetically prepared pre-mates. That is called as rhesus monkeys, called as Andy, A-N-D-I, for incest DNA spelled backwards on October 2, 2000. Sometimes they may ask the questions. The rhesus monkey is also, what is that? When it will be produced, October 2, 2000. Anti-containing jellyfish gene for fluorescent that glows green in the dark. So after that injections, how it will be isolated? The gene is responsible for the jellyfish. See here, you must note it down. Very, very important point. I will show you how it will be. See this one. Say, take an example. So two sample is very, very important. One is called as jellyfish, isn't it? First one is called as, okay, jellyfish. Already you know about the jellyfish look like a structure like this, isn't it? Jellyfish look like a structure like this, isn't it? This is a jellyfish. Inside there is one of the extra chromosomal body present. This is called as what is that? Plasmid. Okay. This plasmid, it has a ability to light during dark time, fluorescent color. It produces the fluorescent color. For particularly, the particular gene is responsible for the production of what is that? Fluorescent color. Fluorescent color. Don't forget, my dear student, this part. For example, the particular gene is responsible for the fluorescent color. Fluorescent. Okay, fluorescent. Fluorescent. Imagine fluorescent color in this part. Okay, so this gene is isolated and also what? Introduced into the monkey. Don't forget, my dear student. Introduced into the rhesus monkey, RH monkey. Already we discussed it. Which one you know? RH. Okay, monkey. Okay, see here. RH monkey. Okay. Okay, monkey. Okay, here you just imagine RH monkey, isn't it? Just imagine this is considered as a RH monkey. Here the DNA molecules also isolated from this one, isolated the this one part, this part also isolated human genes. Okay, this is the human rhesus monkey gene. This one is called as what is the jellyfish plasmid gene. These two are connected together. As a result, the formation of which one you know? You will get it like this, isn't it? Both the gene it will be present. Okay. It introduced the E. coli, introduced into the E. coli. What happened, you know? So as a result, the fluorescent component gene present inside, it's also multiplied and also transferred to the monkey. What happened, you know, during the night time, it will fluorescent color will be shining. That is the condition. It is a new one, isn't it? So that is also a important, my dear students. Don't forget. Okay. See this. Okay. See that. The gene is responsible, anti-containing jellyfish gene for fluorescent. Jellyfish color, fluorescent color. The glow green in the dark. Okay. This is very, very important, my dear. So see that. This is the slide. First GM primate. Will human be next? Human, some of the human beings, suppose we introduce into the human being, what happened, you know? Human is also sometimes night time, what happened, you know? The fluorescent color will be emitted. That is very, very important, my dear. See the next slide, you can understand the structures. See this is the primate. During the night time, what happened? This gene is, this fluorescent color gene is responsible for the, which one, where it isolated, you know, jellyfish. Jellyfish crossed with the which one, you know, rhesus monkey. Okay. After that, what happened, you know, the monkey is also blooming at the night. That is very, very important, my dear student. This is also one of the miracle we will did. We did in the biotechnology, genetic engineering. Likewise, there are so many animals. Likewise, tobacco plant, and monkey, fish, and also even taken examples of another animal, pig also. Okay, so many animals have been reported. So many animals also identified by this technology. Okay, and then next one is transgenic fish. We will finish it up to this level only, my dear students, today. Transgenic fish. The transgenic fish is a super fish of many species such as a common crab, catfish, salmon, goldfish. Okay, these are the example has given. Okay, salmon, goldfish have been produced in commercially oriented. Already you know about, there are so many. It's aquaculture. We used, you know, goldfish. But these are all are produced by the which, you know, transgenic fish only. These are all are considered getting known as transgenic fish. They containing human growth hormone genes. They containing human growth hormone genes. This is also very, very important. Okay, which gene is it? They attain a size twice of that so by non-transgenic fish. Already, you know, it is also used for the, which one, you know, size. According to the size is a very, very important for the commercial purpose. Take an example of fish. The large size means it has a large weight of responsibility. Okay, directly proportional to the size and weight. That is very, very important, my dear students. This is also done by the transgenic fish. Take an examples. So this is an example. This is the ozone port and Chinook, salmon. Gene is isolated. The growth hormone gene is also incorporated, and also it's also inserted to the egg and also replaced to the Atlantic salmon. As a result, we will get the large size of fish for the commercial purpose. Okay, so we will. So this is the structure. So this is the normal one. This is the after transgenic. We'll get it. Okay. So remaining things we will see the next classes, my dear students. Remaining things we'll see the next classes. I will summarize all those things already we discussed. What are the things you know? First of all, you should denote what is meant by insulin. Okay. So what is by insulin? So who discovered? What is my pro-insulin? How many amino acids present in A chain, B chain, C chain? How it will be joined together with the help of the disulfide bridge? Okay. All those things already you know about. Who discovered this one? In the year 1983, Eli Lilly in American company prepared 2 DNA sequences, both are separately A and B. After the separation, it will united together with the help of disulfide bridge. Okay, which plasmid responsible for the insulin productions? Okay, PUC, where it will be isolated Escherichia coli. Okay, that is very, very important, my dear students. And also used for the genetic engineering vaccination process. Maximum, the first one is hepatitis B vaccine. Okay, how it will be already you know about the hepatitis B virus. We have to introduce into the gene to the E. coli bacteria. It's also the recombination technology. As a result, what happens? You will get the 2 hybrid genes present in the single E. coli molecules. Finally, we'll get the vaccination. Likewise, there are so many methodologies have been reported. Okay, and DNA vaccines have many advantages. What are the advantages? Already we discussed that things also. Transgenic animals, particularly, you must note it on this sector is very, very important. A clinical research and pharmaceutical manufacturing sector, that is also very, very important. The term first introduced by the name of the scientist, that is very, very important. Gordon and Riddle in the year 1981. There are so many animals also produced by the transgenic animals. Okay, what are they? Rats, rabbits, pigs, sheep, and cow, and also fishes. Particularly 9% all are mice. Okay, that things also already we discussed. The super mouse in the year produced in the year 1982. Okay, and then the first transgenic cow, Rosie, produced human protein enriched milk 2.4 kg per liter. That content is called as what is an alpha-lactoalbumin protein molecules. That is also very, very important. And transgenic GB, it is very, very important for the one of the hemophilia factor that is called as anti-hemophilia factor 9th one. Okay, the milk contain this component, so we will not suffer hemophilia B. Okay, and then you know about there is a technique already we discussed these things also. This is a dolly. Okay. Transgenic coat. Transgenic coat is also very, very important. There is a blood clotting dissolving protein. Tissue plasmogen activated, simply called as TPA. It is also very, very important in the NEET point of view. They may ask the question in the explanation aspect. Have been formed. They secrete this protein in the milk. Okay. So this is also very, very important. So already you know about how it will be. And then transgenic cow, it is also very, very important. Cow containing, goat containing for antibacterial human ions binding protein, lactoferrin, and super big. Okay, it is very, very essential for the zero transplantations. The organs also transfer from the pig to human beings. Okay, that thing's also very, very important. Okay, so we will finish it up to this level, my dear students. The remaining things we will see on the next classes. Thank you so much for the participation. Bye. Take care. Good night. Thank you, ma'am.
Thank you, sir.
Okay, ma'am. Thank you.